how I've done this in the past is I've just written it this way, where we're talking about the cell cycle. And cells, when we look at their entire life cycle, have several different phases. We can kind of generally break this down into just two parts. There is the first part called interphase. It has some subdivisions of interphase. But by far, interphase is the dominant part of the life cycle of the cell. 90% of the life cycle of a cell is going to be spent in interphase. And then the other part of the cell cycle is going to be called mitosis. This is really just considered division of the nucleus. We're going to see if you divide the nucleus in a way in which the chromosome number stays intact. That's mitosis. If you divide the nucleus in a way that you half the chromosome numbers, well, then we're talking about meiosis. Well, again, we'll see that on Wednesday. This whole process of dividing the nucleus is going to take about 10% of the life cycle of the cell. And then down here, you can see I just put a little asterisk next to this process of cytokinesis. It's technically doesn't have anything to do with dividing the nucleus. That has already been accomplished in mitosis. So this isn't really part of the cell cycle. It's just dividing the cytoplasm that's left. I have it on the list. But there is another visual way to represent the stages of the cell cycle that will allow me to describe the phases of interphase and maybe what's going on with each of the stages of mitosis. And that's going to take us over to this other diagram. I've tried to put what I have written over here, the stages of the life cycle of the cell, and more of kind of a circular pie chart. And you can see the large red arrow represents the time spent in interphase. It's about 90% of the life cycle of the cell. What I've added here is hours that are going to add up to a single day, 24 hours. We're going to see if you take a 24-hour day and you separate that into the stages of the cell cycle, about 10 hours of that day are going to be spent in the first phase of interphase, what we're going to call the G1 phase. About 9 hours are going to be spent in what we're going to call the S phase, and then about 4 hours are going to be spent in the G2 phase. 23 hours are going to be spent in interphase, and only 1 hour is going to be split into dividing the nucleus, what we're going to call mitosis. For example, at the beginning of this G1 phase, we're going to start with cells. I'm just going to draw a cell that has a nucleus, and I'm just going to draw 3 chromosomes inside this nucleus. And I'm going to put a little centromere in the middle of each of those chromosomes. So again, what we're looking at, if we kind of pull one of those chromosomes out, just like we have on the board, this whole thing is one chromosome. It is an unduplicated chromosome. And in humans, maybe just to put a little note down here, since I'm not going to have enough room to write all of these chromosomes, in humans, we're going to have 23 pairs of chromosomes. Okay, so that's 46 chromosomes total. And at the beginning of the cell cycle, each of these chromosomes are unduplicated. In fact, as we go through this G1 phase, the chromosomes are going to stay unduplicated. I'll just kind of maybe bring this all the way to the end of the G1 phase. At the end of this G1 phase, what we see, again, is a cell that has 3 of these chromosomes, and each of these chromosomes are unduplicated. So what's even going on here in the G1 phase? Let's maybe talk about that. The things that happen here in the G1 phase, the contents of the cell are going to duplicate. So I'll say cell contents duplicate. But I have a little caveat with that. The cell contents duplicate. This includes things like the organelles, but it is going to exclude the DNA. So cell contents divide, like I said, you're going to get more chloroplast if it's a plant cell, more mitochondria if it's an animal cell. Again, the DNA does not yet duplicate, so we're still left with these unduplicated chromosomes at the end of the G1 phase. Sometimes I consider the G1 phase is just when a cell is being a cell. And maybe before we even get too far, we mentioned some of these cells never leave the G1 phase. They have their nucleus with unduplicated chromosomes, and they can spend their entire life kind of in this state that we would call the G0 phase. And it's really only when a cell accumulates enough of these proteins internally, we'll call those cyclin proteins. Sometimes they're considered proteins that accumulate when there's been damage done to a cell. It's just one of the proteins that tell the cell it's now time to make another copy of yourself. If those proteins don't accumulate, then you can have cells that just kind of stay in this post-mitotic stage. So your neurons, your skeletal muscles are mostly in this phase. But skin cells or intestinal cells that need to quickly make another copy of themselves, they go through this G1 phase, accumulate enough of these cyclin proteins, and are going to enter the next phase of the interphase, what we're going to call the S phase. In the S phase, let's just describe what's going on, and then I'll try and draw it visually. The S phase is called the synthesis phase because the DNA gets replicated in this phase. So I'm going to say DNA replication. In addition to the DNA replicating, so that's basically we've doubled the amount of DNA, that also is what allows our chromosomes to become duplicated. So DNA replication and chromosome duplication. We can kind of visualize that by showing the fact that we started with cells that had unduplicated chromosomes. And by the time we get out of this S phase, we're going to be dealing with cells that still have 3 of these chromosomes. But each of these 3 chromosomes are now going to be in this duplicated state. I think it's good to visualize this. So again, we've got 3 centromeres, 3 chromosomes, 3 centromeres, 3 chromosomes. In unduplicated chromosomes, for every chromosome, we can do it this way. 1 chromosome equals 1 chromatid. But that's not the case in these duplicated chromosomes. Every 1 chromosome is going to equal 2 chromatids, these sister chromatids. So what's going on up here, now that we're through the S phase, we're dealing with these duplicated chromosomes. There's our centromere in the middle. We've now got these 2 sister chromatids. This is our duplicated chromosome. Okay, so chromosomes have duplicated in this S phase of the interphase, and that now takes us to the G2 phase. What's going on in the G2 phase? It's only 4 hours long. The main thing that's really going on in the G2 phase is the cell is preparing for mitosis. We're just about to enter mitosis. I'll try and say that 2 ways. I'm going to make 2 bullet points. I'll say it just like it says it in the textbook. I'll say the cell prepares for mitosis, and then I'm going to add to what it's actually doing there. Prepares itself to enter mitosis, and one of the things that it checks for before it enters mitosis is it checks for copying errors. So it checks for errors that just might have happened in this DNA replication process. There are proofreading proteins that have to go back and proofread all the replication that has just occurred. And if there are changes that need to be made, those changes are made before we ever enter this next phase which is mitosis. So we're kind of making our way around our life cycle of the cell. We're about to start mitosis, and you can see we're going to start mitosis just like we entered the G2 phase. You've got cells that have duplicated chromosomes. We got one cell, we've got 3 duplicated chromosomes. These cells with duplicated chromosomes are about to enter mitosis. And just to give you the quick shorthand version, over the span of 1 hour, all that's going to happen in mitosis is these duplicated chromosomes are going to get separated. And you're going to end up with 2 cells, each with their own nuclei, and each containing 3 unduplicated chromosomes. Kind of right back where we started with this process of, we're starting with cells that have unduplicated chromosomes. They stay unduplicated until the cell goes into the S phase, in which point those unduplicated chromosomes all become duplicated chromosomes. All that happens in mitosis is those duplicated chromosomes are going to get separated from one another, and we end up with 2 cells that are identical to our original cell. In fact, I'm still using that same cell that we started with. It looks just like the cell that we created. This whole process that we just kind of skimmed over, mitosis, is going to be subdivided into the 4 phases that you see: prophase, metaphase, anaphase, and telophase. After telophase, you're going to see we have 2 separate nuclei. We will have completely divided the nucleus. Following that, I wrote it in a different color because technically it's not part of mitosis. That is the cytokinesis, which is where we just kind of separate the cytoplasm between those 2 cells. Let's see. You can use the acronym PMAT if you want: prophase, metaphase, anaphase, and telophase, to remember the phases of mitosis. But we need to kind of know more about what's going on in each of those phases, because I might be showing you pictures of them. I might be just describing with words what's going on in the different stages of mitosis. Can everybody read that? Maybe? Okay, I'm gonna add to that, since we only have like 8 slides to go through once we pull the slides back down. What we're about to see are some of the detailed stages of this process of mitosis, and I might be able to use the rest of the board. In fact, I'm going to use some of that space. I'm going to take the rest of the board space and try and use it up. Okay, that's given us a big loop that's going to allow us to investigate these different stages of mitosis. Is everybody ready to dive in? Okay, I'll take that as a yes, and we're just going to jump in to the stages of mitosis, and we're going to start with prophase. Again, I'm not a great artist, but I'll try and put the key points of prophase that we need to be able to recognize. In a good prophase, what we're going to see is, let's put a nucleus in here. We know that the nucleus is going to contain these duplicated chromosomes. I'm going to give myself a little more space. Okay, I've basically drawn the exact same cell that we have left the G2 phase with. So what are some of the differences between the cell that leaves G2 and enters prophase? Let's make those changes to the diagram. First off, this nuclear envelope is going to start to fragment. So we can see a fragmented nuclear envelope. And the other thing, that organelle that we failed to mention before, that we now mention a lot, that organelle that organizes those spindle fibers starts to grow. They have not yet moved to opposite sides of the cell. They have not yet grabbed onto any of those chromosomes. But the fact that we can see those spindle fibers growing, we can see the little centrioles kind of kicking into gear, and we can see chromosomes inside the nucleus that's starting to fall apart. These are all clues that we're looking at a good prophase. Again, so we can see condensed chromosomes. We can see spindle fibers form. So we've got chromosomes have condensed, spindle fibers are forming, and then the last thing that we're seeing is that the nuclear envelope is fracturing. Okay, there's a good prophase. We've got condensed chromosomes, spindle fibers are forming, nuclear envelope is starting to fall apart. This prophase is going to lead into kind of an intermediate step between prophase and this next phase that we're calling metaphase. I'm going to call it pro-metaphase, as we'll see in the textbook. It's kind of considered a late prophase or an early metaphase. Here's what a very late prophase is going to look like. We're going to see a cell whose nuclear envelope is now just fractured into pieces. So we've completely blown up that nuclear envelope, and pieces of that nuclear envelope are now just kind of hovering around in the cytoplasm. We can see these chromosomes are now no longer tethered by any type of nuclear envelope, and they're just kind of freely floating around. The other thing that you can see are these microtubule organizing centers. They're reaching out. Some of them are starting to grab on. I mean, they're not yet organized. They haven't made it to their separate sides of the cell, but they're starting to grab onto some of those chromosomes. This is what we're calling a pro-metaphase. I'll just take us from a pro-metaphase over to an official full-blown metaphase. Full-blown metaphase is usually easy to recognize. Again, we've completely ripped apart the nuclear envelope. So the fragments of that nuclear envelope are floating around somewhere. The dead giveaway that we're looking at metaphase is that the chromosomes are going to be lined up along a midline, this mid-plate of the cell. It's called a metaplate. This metaplate is where those duplicated chromosomes are going to line up on. The other clue that we know that we're in full-blown metaphase is that these centrioles are going to be at opposite sides of the cell. And we're going to see that they're reaching out and they're grabbing onto those centromeres from both sides. Look at how those centromeres are now, they have spindle fibers attached to them from either side. So maybe let's add those details about metaphase. We got chromosomes align on the metaplate. So chromosomes align on metaplate. We can see spindle fibers attach to both sides of the centromere. Chromosomes have lined up in the middle. We can see spindle fibers are attaching to both sides of the centromere. And there's something about that earlier statement. We said, as soon as spindle fibers reach out and touch those centromeres, we actually know there's a little Pac-Man protein waiting right there for those spindle fibers to adhere to them. And as soon as that happens, we know those kinetochore proteins are going to start to chomp down and shorten those particular spindle fibers. I'll leave that right there. There's one more thing I want to add to it, but I'll add it here on the next step, which is following metaphase, we're going to see those duplicated chromosomes are just going to separate from one another. That's another easy to recognize stage of the cell cycle. It's going to look something like this. I'll put that metaplate line in there again, except now we're going to be talking about anaphase. What makes this anaphase, we can see centrioles, once again at the opposite sides of the cell. But in anaphase, we have separation of those duplicated chromosomes. I'm going to draw half of this chromosome being pulled back to one pole, and then the other half of that duplicated chromosome is getting pulled back to its opposite pole. We've got spindle fibers that are dragging these things back towards their opposite poles. Here's that other point that I wanted to make. Over here on the metaphase line, I've drawn spindle fibers that are just attaching to the centromeres. And that is true, they do that. And the ones that do attach to those centromeres shorten, just like I've drawn on anaphase. And it's the shortening of those centromeres that touch that kinetochore protein that has now drugged these two duplicated chromosomes in opposite directions. But here's another thing. There are additional spindle fibers that don't ever touch a chromosome. Here are spindle fibers coming from opposite sides of the cell that are attaching not to chromosomes but to other spindle fibers. Deacon, can you do me a favor real quick? My hand is acting like a spindle fiber that is growing towards you, and if you'll put your hand out towards me, you're acting as a spindle fiber growing towards me. We're like centrioles that are on opposite ends of the cell. As our microtubules continue to grow, what's going to happen is I'm going to start to get pushed further away from you back into this door, and you're going to get pushed back into that wall. It's these microtubules that continue to grow. They actually push these poles further apart. So I just want to make that point that there are spindle fibers that are shortening. Those are the ones that are connected to the kinetochore proteins. And then there are also spindle fibers that continue to elongate here in the anaphase. And it's those ones that continue to elongate, they push these poles further apart. So let's add those two things. In anaphase, we've got chromosomes that separate. So chromosomes separate, and then I'll say the centrioles move further apart. All right, and it's those growing microtubules that push those centrioles further apart. So you've got centrioles getting further away from one another, and as those get further away from one another, you've got those other shortening spindle fibers that are bringing those unduplicated chromosomes now to their opposite poles. With just enough room left on here, that takes me into this last phase that I'll draw a cell that kind of looks like a number 8. But what is happening, this is what we will describe as telophase. In telophase, what we can now see is that the unduplicated chromosomes have now made it far enough away from one another that they are now considered to be at separate ends of the cell. Let me put the centromeres in there. We've now gone back to our unduplicated chromosomes, and now those little 2 packages of DNA are far enough apart from one another that those little fragments of the nuclear envelope that we busted apart in prophase, they never went anywhere. They were just hanging around in the cytoplasm. We didn't draw them for a couple of steps, but now the fragments of those nuclear envelopes are coming back, and this time they're forming 2 separate nuclei. So kind of that question from the earlier group is still true. Like, where does the nuclear envelope come from in these 2 new cells? Well, it's actually the same nuclear envelope that was in the original cell. It just fragmented and now has come back together around 2 separate groups of chromosomes, making 2 separate nuclei, indicating that we are now through with mitosis because we've separated the nucleus. We now have 2 separate nuclei. The only final step is just to separate that cytoplasm, this process that we're going to call cytokinesis. So after telophase, we've got this process of cytokinesis. And again, as this cytoplasm gets cleaved into two cells, we end up with cells that are just like the original cell that went into this stage of the cell cycle. Hopefully you can read that. Maybe with that first layer down, we can go back and consult some of these slides to see if that makes sense. Everybody good with that?